Okay, cool. So today I'm going to talk about the genetics of substance use disorders. So my background is that I thought I wanted to be a forensic psychologist, like the people in Criminal Minds. So I went to undergrad for psychology and then realized that everyone had a different theory about how the mind works, and I wanted something more concrete. So then I added a behavioral neuroscience major, and then I got my PhD at Johns Hopkins in human genetics and molecular biology. I did my dissertation on schizophrenia genetics, so using gene editing and stem cell modeling and RNA sequencing to look at GWAS variants and see what they're doing in cells. And then now I'm here working with Rachel, and our lab looks at doing genetic studies of mood and substance use disorders in diverse populations. And Zila just walked in. We all just introduced ourselves. You want to introduce yourself?
Hi, I'm Theo. I work with MD and I do similar things to what Kyra does.
Okay. And just because we tend to be a little bit quiet and shy in this office, I thought I would show you the faces of our lab group. So if you see us, you can be like, hey, I recognize that face, and you can feel comfortable chatting with us if you'd like. So you all know Emily and Zeal, who just introduced herself. So they're at the Hartwell lab. I highlighted Zeal in blue because she kind of jumps around and works for everybody, works with everybody. And then it sounds like you all know Hank. So Sylvanus is postdoc with him, and then also Sylvanus works with Rachel as well. Yusuf is a medical student, Crystal, who was just here, is postdoc, and Jackson's an undergrad. And then the Kember Lab consists of Rachel, who's on maternity leave, myself, Michaela, and Regan, who's our other postdoc. So if you see us around, feel free to say hi. Okay, so I'm sure you guys have probably heard this already in the series about SUD definitions. Okay, so a substance use disorder is characterized by the uncontrolled use of a substance despite it having negative consequences. And I found this cool graphic that has this interesting acronym for the different criteria, which is chew that cop. So there are 11 different criteria for having a substance use disorder. If you have at least two of them, you're considered to have an SUD, and they range from being unable to stop using the SUD even though you try, even though it's having adverse physical and social and vocational effects, having tolerance or withdrawal, um, giving up large amounts of time to obtain the drug or do other things related to the drug, stuff like that. So with the DSM-5 definitions, now if you have 2 to 3 of these criteria, you're considered to have a mild SUD. If you have 4 to 5, you have a moderate SUD, and if you have 6 or more, you have a severe SUD. And currently there are 10 different substances which have an SUD classification in DSM-5. So alcohol, stimulants, including cocaine, caffeine, cannabis, hallucinogens, fiends, cyclines, inhalants, tobacco, opioids, sedatives, and others. Most of what I'll talk about today will be related to these four. So, as you might imagine, alcohol and tobacco are the substances for which we have the most data because they are legal and therefore the most commonly used. Cannabis is now becoming legal in some states. We're getting some more information on it, and opioids are obviously still illicit, but because they cause such intense problems, there have been a number of studies on it. So what causes SUDs? So in our field, we call SUDs, refer to them as complex. And that means that they are conditions that are caused by a combination of both genetic and environmental causes. So there are a number of known environmental factors which can contribute to SUD development. These are just a few of them. These can include socioeconomic status, whether or not your peers are engaging in substance use, and traumatic experiences. There are known behavioral factors, which can include starting to use a substance early, like early in life, and then using it at a high frequency early in life, as well as having emotional issues during your childhood with both internalizing feelings and externalizing feelings. Obviously, there are also genetic factors, which I will talk about more. And then importantly, there are interactions between all of these things. And we might think about family history as a good example of this because your family history is based on genetics, whether or not other people in your family have an SUD, but then also it can be environmental because if someone around you in your family is using, then that can influence your decision to use. Hi. Did you want to introduce yourself? We all kind of just like, sorry, I didn't mean to like call you out, but
My name's Astrid. I work with current. I'm a rising fourth year PhD student.
thank you.
Yeah.
Yeah, we have a combination of undergrads and PhD students and research coordinators and people in here, so you've right in. Okay, so we're obviously going to focus on genetics in this talk. So how much do genetics really matter for substance use disorders? So when we talk about how much genetics matter for something, we tend to use the term genetic heritability. And the technical definition of genetic heritability is the proportion of phenotypic variance that is explained by genetic factors. An easy way to think about this is the amount to which genetics typically contributes to SUDs in a given population. So we know from twin studies that the genetic heritability of SUDs ranges somewhere between 50 and 70%. So here I'm showing you this graph, and on the y-axis is the amount of phenotypic variance explained, so the amount of variability we see in whether or not people have SUDs. And on the y-axis are different measures of genetic heritability. For the moment, I'm going to show you this plot multiple times throughout the talk, but right now I just want you to focus on these 4 bars. These are showing the overall genetic heritability for alcohol use disorder, cannabis use disorder, opioid use disorder, and tobacco use disorder. So you can see, depending on the phenotype, the heritability ranges from about 50% to about 70%. So we know that genetics plays a role in SUDs. So what kinds of genetic factors contribute to SUDs? So when we talk about conditions that are complex, we talk about the things that make up the genetic architecture of those complex conditions. So when we talk about complex conditions, we're talking about how both genetics and environment contribute to that condition, but we're also talking about how there's a mixture of different types of variants that contribute to that condition. So there are both rare and common genetic variants that contribute to SUDs. And this type of plot is something you'll see pretty often in genetics. So here we're just describing how often different types of variants occur based on how much of an effect size they have, like how strong their effect is. So on the x-axis here, we have allele frequency, and on the y-axis, we have effect size. And there are different classes of variants here. So we can see that, so down here will be rare variants, and up here will be common variants. And there are sometimes rare variants that will have a small effect on a phenotype, or rare variants that have a large effect on a phenotype. Or there could be variants that are common but have a high effect, and variants that are common that have a low effect. Most of the time, genetic studies look at these two, so rare variants with a large genetic effect, or common variants with a small genetic effect. We know that SUDs are polygenic, and so most of the influence from genetics comes from common variants with small effect. For the sake of time and because of the bounds of my own expertise, we're going to focus just on 2 today. So the first one will be high effect common variants, which actually are pretty uncommon. And SUDs are unique in that there are a few common variants that have a large effect size. So here's some examples. And these were among the earliest variants that were discovered for SUDs, mainly because they're common. And so that makes them easier to find. They have a larger effect size, which means if you find it, it's most likely to be doing something, so it's easier to find. And also because they lay in genes that are directly related to the metabolism of that specific substance. So if you were like a geneticist back in the day and you're going, hey, what genetic variants could be important for alcohol use disorder, you'd probably look at genes that affect alcohol metabolism, right? So they just like logically make sense. So for OUD, a common gene in which common variants are seen is OPRM1. This gene encodes the mu opioid receptor, which is one of the main receptors that opioids bind to, like fentanyl and heroin. And the discovery of this gene has led to the development of useful clinical medications. So OPRM1 is the target of methadone, buprenorphine, and naltrexone. For AUD, 2 important genes are ALDH2 and ADH1B. These genes both encode proteins that are involved in the metabolism of alcohol. And ALDH2, I believe, is targeted by the drug antabuse. I don't know if you guys have heard of that, but it's a drug that you take and it basically gives you like a really bad reaction to alcohol to sort of dissuade you from drinking. And finally, for tobacco use disorder, 3 well-known genes are those involved in those that encode subunits of nicotinic acetylcholine receptors, which is what nicotine binds to. And CHRNA5 in particular is targeted by Chantix, which I've definitely heard commercials about Chantix, but it's supposed to help you stop smoking. Okay, so those were some genes in which there are common variants with high effect sizes. I'm going to spend the rest of the time talking about common variants with small effect sizes because that's where most of my work is involved. So some people in here aren't super familiar with GWAS, so I'll just do a really quick review of the basic concepts of GWAS. So the point of a GWAS is to find common variants that are associated with a phenotype. So you'll take a group of people who are cases and a group of people who are controls. So for example, people who have alcohol use disorder or do not have alcohol use disorder. You extract DNA from them, and then you get genotype data for those people. And what you're looking for is any variants that are more common in cases versus controls. And so in this example here, we're looking at this red variant here. These are depictions of chromosomes. And we can see here that this particular variant in red is common in 58% of the case population, but only in 16% of the control population. And so we would say that this variant in red here is a risk variant for AUD, if we're looking at AUD. Does that make sense to everyone? That's an important concept. Okay. So to be specific, genome-wide association studies find a locus that's associated with a phenotype, or a locus means a genetic area. So it does not find a specific variant that is causal for a phenotype. It finds a group of variants because some variants are inherited together in a pattern. And so when we're looking at the results of a genome-wide association study, we visualize them like this in this type of plot, which is called a Manhattan plot. And the way we read this is on the x-axis is the genome shown by the chromosomes. So it's the base pair position in the chromosome. And on the y-axis is the significance statistic. And any variants that are crossing this dotted line here are considered to be genome-wide significant, meaning that they are statistically associated with whatever phenotype you're looking at. So in our AUD case, this would mean that these loci here are associated with AUD. And you'll notice that there are these sort of peaks here for each of the significant variants. And this is the reason why it's called a Manhattan plot, because they look like skyscrapers kind of. And the reason that that pattern happens is that all of these variants are in what's called linkage disequilibrium. So they're inherited together. And so each peak is telling you that, hey, the variants in this locus are associated with AUD. So we know that something in this locus here is causing the association, but we don't know which one in this locus. So each of these peaks is tagging a locus.
When does it becomes important? Is it after it passes that like dash line that we start paying attention to?
Yes, exactly.
Okay.
Yeah, so this dash line is our arbitrary statistical significance threshold that we use to account for the fact we're doing so many tests across the genome. Yeah, sometimes people will play around with this threshold depending on the question they're asking, but most of the time it is 5 times 10 to the negative 8th. Okay. It's important to know what GWAS is telling us and not telling us. So it does tell us that something in that locus is somehow involved in the phenotype. And it does that specifically by giving us a beta value for the SNPs in that locus. And beta just tells you how strongly that SNP is associated with the locus. So if the SNP was in every single person who was a case, but not any of the controls, we'd say, okay, this SNP really is associated with AUD. In that case, it would have a really high beta. If it's only a mild association, so sometimes people have this SNP if they have AUD, but not all the time, then it would be a lower beta. And that beta value is what we use for downstream analyses. But importantly, GWAS does not tell us a specific variant or a specific gene that is causing the phenotype. This is just seeing a relationship of them happening at the same time. We don't know for sure that this is directly causing this. You would have to do downstream analyses later in order to determine that. But what it does tell us is there is this region of the genome, there are certain variants in that region, those could be important. Okay, does that make sense? Forgot to start my timers. Okay, so now going back to GWAS in context of SUDs. So because most of the variants underlying SUDs are common, and that means they have a small effect size. So in order to get enough statistical power to find the association between these variants of small effect size and SUDs, we have to have really big sample sizes, like millions of people. Not quite millions, but we're aiming for the millions now. And in order to do that, to get these massive sample sizes, we have started using something called electronic health record biobanks. So like in a traditional genetic study, in a traditional GWAS, we would want to get a group of people who are cases and a group of people who are controls. And so we would go, hey, we have to find a bunch of these people who have AUD and a bunch of people who are healthy. So you would end up like recruiting people, posting flyers, trying to find participants for these studies. And that was good because it gave you a really clean set of cases and a really clean set of controls, because you could be like, I'm only enrolling this person if they have XYZ criteria. But the problem is that it costs time and money to recruit people like that. So an approach that a lot of people are taking now is using biobanks that exist at different hospital systems like Penn, and using biobanks that have linked electronic health records. So when you go to the doctor, all of your information gets recorded in an electronic health record. Some hospital systems have a biobank where you can consent to have your genetic information and your EHR data linked together and then de-identified, and then researchers can view it and use it to do genetic studies. So a number of academic hospitals around the country have biobanks. Penn is one of them. And so the Penn biobank is called PMBB, Penn Medicine Biobank. There are also a number of other large US-based biobanks that are important for these GWAS because they have huge sample sizes. So one of them is the Million Veterans Program, which is run by the VA, which Emily and Zeal, who they work for. And then also and that's a population of now 1 million US veterans who have consented to have their information shared for research. And then another one is called All of Us, which is a US-based biobank that recruits people from different hospital systems across the country. And the focus of All of Us is really on being really diverse. So having people who are not just of European ancestry, but also African and Asian, so on and so forth. So those are some examples of biobanks, EHR biobanks. So there are dozens and dozens of biobanks. And so we're all working together in these consortiums to do genome-wide association studies together. So two of the consortiums, for example, that our group is really involved in is the PsychEmerge Consortium, which is a group that's focused on looking at psychiatric and substance use conditions, and then the Psychiatric Genomics Consortium, which is about like 80 different cohorts in a whole bunch of different countries, whereas PsychEmerge is US-based. Okay, so I told you about how to run a GWAS. So there are multiple ways in which people have approached doing GWAS for substance use disorders. So to start sort of basic, you can do a GWAS looking at substance use disorders. So this would be based on whether someone is a case or a control for a substance use disorder. Doing a GWAS based on case control status of an SUD is useful because in real life, that's what we're seeing in the clinic, right? People come in, they get a diagnosis for AUD. We want to know what genetics could underlie people getting diagnosed with AUD. Perhaps one downside of doing it like that is you have to have a diagnosis, and not everyone has access to a clinic where they can get a diagnosis from a clinician. So in some cases, this can reduce our sample size. Another way you can approach it is instead of looking as a case control, you can look at it in terms of use or symptoms. So for example, instead of asking, does this person have an alcohol use disorder? You can ask how many drinks per week do they have? How much alcohol do they consume? Or for tobacco, how many cigarettes do they smoke per week? So this is getting at use patterns rather than a diagnosis. And this is useful because it can capture patterns that are shared across multiple SUDs because we know that they tend to be comorbid. And also because of statistics and statistics black magic, if you use a continuous variable instead of a binary variable, you have a lot more statistical power for your analysis. Also, I don't think I put a bullet point here, but this can be captured based on self-report. It's like in a survey, so people wouldn't necessarily have to come into the clinic and get a diagnosis for that. So here are a few tables that summarize some of the SUD GWAS that have been run. I apologize for the small text, but essentially what I'm just showing you is that here are a whole bunch of GWAS that have been run for alcohol use. You can see here this study, which came out, I think it was last year, had almost 3 million participants. This is looking at drinks per week, and they found 496 significant loci associated with drinks per week with alcohol consumption, which is amazing. And then here, this was like an AUD-based phenotype, and here they had over 1 million people, and they found 90 different loci. And then here's a similar table for cannabis and tobacco. So in 2023, there was a paper looking at about 1 million people with cannabis use disorder. They found 26 loci in their largest sample. And then here's for tobacco. Here's a study of over 3 million people looking at smoking initiation. They found 1,346 loci. It might just be like the biggest result ever from any GWAS, maybe besides like height or BMI or something. And then here's tobacco use disorder, which was done by our very own Sylvanas, whose office is right down the hall if you want to chat with him. And he found 88 loci. And then finally, here's opioid use disorder. You'll notice from this that the number of significant loci is much lower because it's harder to get samples on opioid use disorder because it's illicit. So this drug is definitely lagging in research behind others. Okay, so we can do GWAS for SUD level analyses. We can do GWAS for use level analyses. Another way some people are approaching it is comparing the two. So doing a GWAS stratified by whether you have an SUD diagnosis or you just use a substance. And the point of doing that is it can find the difference in genetic risk between those who use a substance versus those who actually become addicted to the substance. So someone who just drinks a lot because they feel like it versus someone who drinks a lot because they can't control themselves. And our very own Rachel Kember did a study on this a couple years ago. And here's her Manhattan plot. And on the top here is the GWAS she ran for people who have an AUD diagnosis. And she found 33 loci. And on the bottom here are people who were not addicted, but did consume high amounts of alcohol. So it's consumption. And you can see that she found 33 for AUD and 30 for consumption. And if you look at the location of the peaks here, you can see the differences. So like, for example, this genetic variant here is associated with consumption, but not with addiction. And so narrowing in on some of these genetic differences could help us understand why some people who are able to consume substances without becoming addicted, so that might lead to therapeutic candidates. Another really cool and really cutting edge method that people are using is something called genomic structural equation modeling, which Crystal Davis is an expert on, and she shares an office with me down the hall. So just so I don't confuse you, just like the very basic idea of GSEM is that it lets you take summary statistics from a trait it lets you find common genetic variants that are shared across multiple traits. So what you do is you take GWAS results from traits that you care about. So let's say 3 different SUD phenotypes, so like AUD, OUD, and TUD. You do some fancy math, and you're able to create this unobserved common factor that just represents all the shared genetics between those. So you might imagine for SUDs, what's in common between SUDs? Addiction, right? Alcohol genetics might include, like AUD related genetics might include stuff related to alcohol metabolism. And TUD genetics might include stuff related to tobacco metabolism. But the thing that's shared across those is addiction, the genetics of addiction. And so once you have this common factor, you can then do genetic studies on that to ask, okay, well, what genetic variants are underlying, what specific genetic variants are underlying the shared genetic stuff between these different SUDs. And so people have done this. And so one study was done in 2023 where they did exactly what I just said for addiction. So they used GWAS summary statistics for AUD, CUD, OUD, tobacco use, and then cocaine use disorder. And they made what they called the addiction factor. And then they ran a GWAS on it. And here's the result of that. And what they found was that there were 17 loci associated with this generalized risk of addiction. And they identified specific loci that were tied to it. They did some downstream analyses and predicted that these variants were involved in brain dopaminergic signaling, which makes sense because dopamine is involved in reward pathways. And so it makes sense that that would be linked to addiction. This is a really cool study. This was a study looking at the externalizing factor. So externalizing, if you're in psychology, you probably know what externalizing versus internalizing is, but they're ways of interacting with the world. So like internalizing traits are traits related to how when you experience feelings, you kind of like keep them to yourself. I'm definitely not explaining this the proper way, but then externalizing are behaviors that are when you react to things happening to you, you react to them externally. So for example, when they were generating an externalizing factor, the types of things they fed into that were number of sexual partners, smoking initiation, alcohol consumption, ADHD, cannabis use, age of first sexual encounter, and general risk taking. And the reason they did this is because it's known that substance use is associated with generally like risk taking and risky behaviors. And when they created this externalizing factor and did a GWAS on it, they found 579 loci and found that across these loci, there was a common link to neurodevelopment. Okay. So another thing that people do, which is kind of an active area of research right now, is doing sex stratified analyses to find differences in genetic risk between males and females. Not a lot has been done here because females tend to be underrepresented in SUD studies. So if we go back to these tables I have here, this column here, I don't know if you can see it, but this column here is showing the percent female of the participants in that study, and most of them are under 20%, except for OUD papers, or it's just not reported. So doing sex stratified analyses is something that's being actively done right now. Rachel has a grant in to look at differences in alcohol use disorder. I'm actively working on a project with the Million Veterans Program to do a sex stratified study of tobacco use disorder. These are some of my preliminary results. So on the top here, I have my Manhattan plot for tobacco use disorder in males, and on the bottom, we have it in females. Something that immediately strikes you is that this is the genome-wide significance line in males, and there's a ton of hits. And down here, we don't have a genome-wide significance line because none of them reached it. And you'll notice that that's probably because the sample size for females is less than 10% of that for males. So this is an ongoing problem. A big reason for this is that in MVP, this is pervasive because most US veterans are male. But because MVP makes up such a large proportion of GWAS for SUDs, that means that the GWAS will have fewer males than females, fewer females than males. But this is actively trying to be fixed. Okay, So then once you've calculated a GWAS, then what do you do? So a very common thing to do, and what our lab does a lot of, is calculating polygenic risk scores, or PRS. So PRS are a summary of a person's genetic risk for a trait. Sometimes they're also called a polygenic score, PGI, PGS, or a polygenic index, PGI. And they are calculated as the sum of a person's genetic variants. And then each variant is weighted by that effect size beta that we talked about earlier from the GWAS. So if you're a visual person like me, this will help.
Sorry.
You're good, you're good. So just in this example here of how we would calculate a PRS, let's say we have all these different people, and these are their genotypes at different areas in the genome. And at each spot where it says a V, that person has a genetic variant that has been associated, let's say, with AUD. And we know from our GWAS of AUD that these variants have a beta value of 1 with AUD, and that these variants have a beta value of 0.5 for AUD. So just what you do is you go across the genome and you add up the variants. So this person has 2 variants that have a beta of 1 and a beta of 0.5. So their PRS is 2.5. And then this person has one of 1 and 0.5, so it's 1.5. So it's quite simple. So why do we want these? PRS are useful because they can help us predict who's at risk for developing an SUD. That's the ultimate goal. The ultimate goal is precision medicine, where someone can come into the clinic and be like, hey, here's my genes, like take some of my blood, look at my genes. What am I at risk of developing? And the benefit of a promising benefit of PRS is that because they're based on genetics and your DNA is always the same across your whole life, they can be calculated super early in life before SUDs develop. So we talked about earlier how using substances as a teenager or in early adulthood is a big risk factor for developing SUDs. So if you can have your PRS calculated before that happens and you grow up knowing, hey, I have an extra risk for developing this if I use this stuff, I should probably not do that, then that can help prevent SUDs. And also because DNA is the same across every cell type in the body, that means you can calculate PRS from like a blood sample, which is super easy to get. People might be coming into the clinic to do a blood draw anyway, so it's non-invasive, it's cheap, it's easy. And so also PRS allows us to bin people into high and low risk groups. So most people will have a PRS that's sort of in the middle. It tends to follow a normal shaped curve. And we might want to really focus on the people who have high risk for developing a PRS for risk prediction and for like predicting it in the clinic and preventing, what's the word, preventative care. And we also might want to focus on the people who are low risk and be like, hey, what's going on with you? And why are you so low risk? And what can we learn from that? Okay. So we talked about heritability and how genetics contributes to heritability and how GWAS can help us find genetic variants that explain heritability. However, something that's funny is that when we look at the heritability that's explained by the variants we see in GWAS, they don't account for everything. So here I'm showing you this graph again, and now what I'd like you to focus on is the difference between the height in this bar and these bars across these 4 phenotypes. So in this case, H squared, again, is our total genetic heritability, so the total amount to which genetics contributes to AUD, OUD. And then H squared SNP here is the heritability, the proportion of that's accounted for by the SNPs we find in GWAS. And then PRS-R squared is the proportion of that's captured by our PRS that we generate from the GWAS. And what's obvious here is that they each get progressively smaller. So what this is saying is that the variants that we're finding, like the genetic signal we're finding in GWAS, is like some of it's missing. Like if we look at this here and look at this here, like this whole part's missing. So like where is it? What's happening? And this is called missing heritability. And we know that there are many sources of missing heritability. So like we have some idea about what's going on, But each of these things that I'll talk about is like an active field of research right now. So we know that missing heritability can come from a number of things. One thing would be starting at the very beginning when we get our cases and our controls for our GWAS. How do we define cases and controls? Are people giving our providers giving an incorrect diagnosis to someone? We know that there is provider bias, especially for people of different races. So for example, I think people who are African American tend to be diagnosed with schizophrenia more often than people who are not African American. And I think for OUDs, it's the opposite direction. Remember?
Well, I can tell you an alcohol example that
Mhm.
Rachel Vicker-Smith, who used to be here, who's now back to University of Kentucky, did in the MVP sample, where she found that African American men were diagnosed with alcohol use disorder at lower levels of consumption compared to the European male counterpart. Had to be drinking more to get the AUD diagnosis. It's interesting.
Yeah. So you imagine if that's happening, first of all, that's going to mean that more people who are men are going to be diagnosed as having AUD when maybe they don't.
Mhm.
And so they'll be included as a case in an AUD GWAS when they really shouldn't be. So that can like add noise to your case population and add noise to the genetic findings from that. We also have to account for the fact that many of these SUDs are comorbid. And so someone might come in with a symptom that fits multiple SUDs. And so they might get the incorrect diagnosis. Also, I talked about how we work with consortiums. And so that means that there are multiple sites that are contributing data. And the way in which they define cases and controls at each site might be different. So one common way we do things in our consortium is we say, in order to be considered a case, you have to have 2 instances of being diagnosed with an SUD. Well, at another place, they might say, okay, you just need 1 diagnosis code, or you might need 2 outpatient diagnosis codes and 1 inpatient diagnosis code. So the way in which you define the cases and controls can add additional noise to your case and control sample. Another, like, sort of like moving down the flow of a GWAS, we talked about defining our case control populations, and then like, what about the actual genetics of it, right? So the types of variants that you're looking at in a GWAS. So when we look at a GWAS, we're looking at common variants that are single nucleotide polymorphism, so single base pair changes. But there are many other types of genetic variation. We just talked about before how there are we also talked about before how on that chart there were rare variants with small or large effects. So we don't look at those in a GWAS because the design of a GWAS precludes us from doing that. GWAS by definition are going to look at common variation. So for example, copper number variants are another type of genetic variation that's not looked at in a GWAS. So those will be like multiple base pair changes as opposed to single base pair changes. And so we know that like we're missing some stuff because we're not looking at it in a GWAS. We're not looking at other types of genetic variants. And then we have to account for the fact that genes don't happen, like genetics doesn't happen by itself and environment doesn't happen by itself. They interact with each other. And so things that we talked about earlier, like socioeconomic status, peer substance use, and traumatic experiences can have an impact on your genetics and your epigenetics and how that influences your risk of SUDs. And just to drive that point home, this is the slide from earlier, like outlining some of these factors. We have to account for the fact that they interact. Another thing we have to think about, which I've touched on a little bit, is that SUDs are comorbid with other psychiatric traits and other SUDs. I don't think it would be an exaggeration to say most of the time if you have an SUD, you also have something else like depression or anxiety. I think that would be the norm rather than the exception. Would you agree with that, Emily? Yeah. And so this is called a forest plot. It is showing it's showing the genetic correlation between different substance use traits and other phenotypes. So the genetic correlation is just like a measure of how similar the responsible genetics are between different phenotypes. So like how much of the genetics are shared, sort of like that. And so anything that's over this 0 line is showing that the genetics for those 2 phenotypes are related. So in the different colors here, we have different types of substances, and we can see that for a number of different psychiatric traits, neurodegenerative traits, And socioeconomic traits, if you like the genetics overlap between substance use disorders and those things. So if you're at risk for one, you're at risk for another genetically, is what this is saying. So some of these include like Alzheimer's, anorexia, depression, neuroticism, schizophrenia. So yes, they're highly comorbid. And here you'll just see that these are negative because this is saying if you use substances, you're less likely to finish college. And if you use substances, you're likely to have a lower childhood IQ. That's why they're switched. Okay. So looking at interactions between all of this stuff is basically where the field is right now. So there have already been like a whole bunch of big GWAS, like I talked about here, that have looked at the SUDs in isolation. And this is like a continually rolling machine. So the consortiums that do these are just like continuously collecting more and more and more data. And every few years they put out a new paper with like new results with more diverse samples, more stringent definitions, stuff like that. But kind of like the hot area right now is looking at all the interactions with all this other stuff. So not to be biased at all, but I'll just give you a quick peek at stuff that our group has been doing here. So I was surprised when I came here 2 years ago to learn that like the people in this department are like powerhouses for genetic studies of SUDs. If you look at pretty much any paper for an SUD GWAS, you will see Rachel's name on it or Hank's name on it. And a lot of things, Emily's name on it too. On the more phenotype side, I guess. Yeah. Okay. So stuff that we're working on that has to do with SUD comorbidities. Rachel and Emily put out a paper a few years ago that looked at the comorbidity between SUDs and a wide range of different phenotypes using a technique called phenome-wide association study, or PheWAS. And essentially what PheWAS does is you take your polygenic risk score for an SUD, so let's say AUD, and again, your polygenic risk score is just like a number that summarizes how much at risk you are for that thing. So if you have a high polygenic risk score for AUD, you're very likely to get AUD. And they asked, okay, well, if you have a PRS of this score, how likely are you to also have other stuff, a whole bunch of other stuff? And here's just an example figure from their paper. So these are all the different phenotypes they looked at. And this is the significance. So stuff that's to the right was stuff to the right are phenotypes that were more strongly associated with genetic risk for different SUDs. So you can see like environmental things popping up like household income. You can see trauma, other types of substance use, and then behavioral stuff like shoplifting, stealing, and then also more like environmental exposure to traumatic events. So really driving home the point that it's not only that these things happen together, it's that genetically they are linked. Yusuf, who's a med student working with Hank and Crystal, and also all of us, but they led this paper, did a really cool paper last year, which got published earlier this year, that was using genomic structural equation modeling, which I touched on earlier, to look at the shared and unique genetics of substance use disorders, mood disorders, and psychosis. So asking what genetics are shared between mood disorders, psychosis, and substance use disorders, and what genetics are unique to each of those. And that's a really cool paper. I would look at it if I were you. And then here's just a Manhattan plot showing the results from each of their factors. This was looking at psychosis, this was looking at mood and anxiety disorders, and this was looking at substance use disorders. And not only did they find a ton of hits for each of these, they also did something called GWAS by subtraction, which essentially was looking at the unique genetics that were not shared between these things for each of those phenotypes, and they got significant hits for those too. So that was like helping to try to unpack the biological mechanisms that are specific to each phenotype. And then our own Zeal and Emily are currently working on a paper looking at interaction between alcohol use disorder and major depression. And so they did a GWAS where they took people who had alcohol use disorder and also had depression, and then took people who have alcohol use disorder but do not have depression, and then compare the two. So here you can see, this is technically called a Hudson plot because it's like a reflection of Manhattan on the Hudson. It's like, yeah.
It's called the Miami plot.
Miami plot? Yeah. Is there a difference between Miami and Hudson?
I don't know.
Yeah.
I have no idea.
Okay. Yeah, but you can see clearly here that there are differences in the genetic architecture of people with or without major depression who have AUD. So looking at the interactions between them. Besides other psych and behavioral traits, we also are looking at other types of phenotypes that are comorbid with SUDs. So Sylvanus, this has become his signature thing he's into, his pain. And he has done a large GWAS on chronic pain, and he found significant genetic correlations between chronic pain and substance use. So this is another genetic correlation plot, a slightly different format, but here now on the x-axis is the genetic correlation. Anything over this line here is correlated, and then here's the significance. And anything that's in this like pink color here are substances. So you're just seeing here that the genetics between substance use disorders and pain overlap. And then we also know that SUDs happen together with other SUDs. So if you use tobacco or alcohol, there's a good likelihood you also use tobacco or alcohol or other things. So something that myself and Rachel are working on are trying to develop more precise polygenic risk score models that that can use the addiction factor that I talked about earlier, which is like a general index of addiction risk, and also the externalizing factor to try to see how well each of those predict different SUDs. And something that's really interesting is that on the left side here, I'm showing the ability of the polygenic risk scores, like how strong the polygenic risk scores are, like their link is with the phenotype. So this is in European people and this is in African people. And then this is like alcohol phenotype, cannabis, tobacco, opioid. And the dots here are showing the ability of the polygenic risk score to predict the phenotype. So if you have a higher value here, it means you're more likely to have the phenotype. And so the first dot here in each plot is showing the PRS for that matching phenotype. So like, for example, in AUD, I used an AUD GWAS to make an AUD PRS, and I'm seeing how well that predicts AUD. Then the black dots are showing how well the addiction factor polygenic risk score predicts that phenotype. And the purple is showing how well the externalizing factor polygenic risk score predicts that phenotype. So basically what we see across all of these is that the externalizing factor is really, really good at predicting SUDs, sometimes better than like the actual PRS for that SUD itself. So that's telling us that, oh yeah.
I was just gonna say, what do you make of that?
That something about externalizing behaviors, like in the genetics underlying externalizing behaviors, captures addiction and substance use better than just the substance use. So like there's perhaps the GWAS we have for individual substances isn't capturing like the brain addiction component as well as our genetic studies of externalizing and addiction in general. And then on the right here, this is like a complementary plot showing that R-squared value I talked about, which is like the proportion of genetic heritability that's being captured by this polygenic risk score.
Would you say that the externalizing factor captures both behavior and genetics, whereas the other one like just captures genetics?
It's possible, yeah, because so many of the traits going into externalizing are behavioral. Yeah, good point. Yeah, so this is just a complementary plot showing like how much variance each of those polygenic risk scores explains. And you can see that the externalizing one in certain groups has like 3 times as much variance explaining power. But if you look at the y-axis here, you see it's actually like a very small amount. So this is like 2% of genetic heritability is explained by PRS. So that's definitely an area that still needs to be improved. Reaching the end here. So in addition to looking at other SUD comorbidities, people in our group have been looking at interactions between genetics and environment. So Jackson, who is an undergrad with us, he has looked at the interaction between childhood adversity and substance use and genetic risk. And interestingly, he found that the higher your genetic risk, which is on this axis, the lower the impact of adverse childhood experiences. Oops. And I'm not totally sure what he made of that. I didn't get to read his paper. Yeah, that's ongoing. And finally, my most important point, probably, especially because for some reason, diversity is a hot topic right now, and it's under fire for who knows why. So a huge problem with genome-wide association studies in general, not just for SUDs, is that they are overwhelmingly Eurocentric. So this is a tool that's really cool. It's called the GWAS Diversity Monitor. It's just a website. And if you go there, it updates every day and keeps count of the ancestry makeup of people across all reported genome-wide association studies. So you can see here, as of yesterday, 90% of people who participated in GWAS were of European ancestry. There is a huge effort being made to include more diverse populations, but obviously we still have a lot of work to go. Here's an example of what the breakdown looks like specifically for SUDs. So over here we have alcohol phenotypes, cannabis phenotypes, opioids, tobacco. And we can see that if we look at the color breakdown here, most of these samples are orange, which is European, and it's vastly outnumbering the people of other ancestries. And I'm going to come back to this plot for the last time. And now what I'd like you to focus on is the presence or absence and also relative size of the different colors of these bars. And so what we get from this is that H squared, which is, again, broad genetic heritability, it's the degree to which genetics are contributing to a phenotype. That measure has, for most cases, only been ascertained in people who are European. A lot of these studies happened like there was like a wave of them, I think like in the late 90s and then a bunch in the early 2010s. But we don't even have a metric for what genetic heritability is for SUDs in people who aren't European, at least not large studies. Oops. And then if we look at H-scored SNP, which again is the amount of heritability we're seeing from a GWAS, we can see that in many cases, it's lower for Europeans. And then if we look at polygenic risk scores, in a lot of cases, it's not even measured for people who aren't European. Why do we want to increase diversity in GWAS? Morally, most importantly, it makes the results beneficial to more people. More specifically, it gives us better statistical power when we do ancestry-specific GWAS in groups of people who aren't European, which will let us find genetic variants that specifically act within that ancestry. But also, it's beneficial for everybody because, in general, multi-ancestry GWAS, meaning GWAS that have multiple different ancestry groups in them, tend to find more loci than ancestry-specific GWAS. So if you did the analyses separate, you're not going to find as many results as when you put them together. So it's better for people who are underrepresented, and it's also better for people who aren't underrepresented. So there's no reason not to do it. So I'll finish up with this real-life example of the consequences that happens when you don't consider genetic ancestry. So there's this test called the AVERT-D test, which was approved by the FDA last year. FDA approved. And the point of it is to help predict risk for OUD using common GWAS SNPs. So the idea of this test is it's given to people who are about to have a surgery, and so they'll be in pain. And so this is supposed to help people decide whether or not they should take an opiate to treat that pain to see if they will develop OUD. So based on your genetics. So if you had to take a gander, how many SNPs, how many variants do you think that this test is based on? If you had to guess, like if you had to develop your own genetic test, thinking about how many loci were found for all those other studies, how many would you use in your test? Take a guess. No bad answers.
200.
200. Okay. What about you guys? What do you think? No stupid guesses. I'm not going to move on until someone takes a guess.
I think usually the score would be more reasonable. Maybe
How many variants are usually in a polygenic risk score?
thousands, like 800,000.
Hundreds of thousands, millions. Yeah. It uses 15. And what it does is it feeds these 15 into a black box machine learning algorithm that's proprietary, so they won't tell us what it is. And then they say, okay, we're going to predict OUD risk. Crystal, who's my office mate, was like, nah, and found last year that they don't actually predict OUD risk, they predict genetic ancestry. So if you look here, this is an ROC plot. For those of you who are not familiar with machine learning, basically each line here is an analysis that Crystal did, where she took the 15 SNPs and used a few different types of machine learning algorithms and tried to predict, based on these 15 SNPs, the same SNPs that this test used, if you're going to develop OUD. And this dotted line here is chance. So what you really want is your line to be like way up here like this. You can see that these lines are super close to here, which means that this algorithm basically doesn't do better than chance. These algorithms that she used using the same 15 SNPs that they did doesn't really do better than chance. But when she asked, what do these SNPs predict? She found that they predicted ancestry. So look at the performance of that compared to that. So you can imagine the implications of this. Like, you go into the clinic to get a test to try to help you decide if you should take an opiate, and your result is not because of, yes, you're genetically at risk, it's because you're Black or you're white, and it could be wrong. So this has real world implications. It's really important. So it's not just like a theoretical thing we're talking about, like it's affecting real people right now. So on that cheerful note, to summarize the genetics of SUDs and where we are now, what have we done? There have been numerous studies of substance-specific genetic variants that have led to the development of effective medications for AUD, OUD, and TUD. Actually, right now there's a trial going on for naltrexone that Hank is administering with some really promising results. You might remember that naltrexone targets some of these genes that were identified from these studies. When we look at studies of common variation, GWAS has identified hundreds of risk loci that are currently being explored for their biological function to try to predict which are causal. And we have been able to develop polygenic risk scores that can predict risk for SUDs based on real-world clinical data. What we need to do moving forward is obviously include people who are more genetically diverse, include more biologically female people in genetic studies of SUDs. We need to continue examining the interaction between genetics and environment and behavioral factors and comorbidities. And eventually, you can only do so much with the computer. You have to get to the bench and do wet lab stuff to actually prove which variants at these different GWAS loci are functional and are causal. Thank you for listening. These are my supervisors, and feel free to shoot me an email if you have any questions.